Hello everyone, I am Kakuli Malakar from Northeastern Hill University presenting the poster entitled Synthesis, Characterization and In vitro Anti-Proliferative Assay of Dithiocarbazate based compounds. First, let me show you the structure. That is the structure of the compound. We have designed it systematically. You can see that first we introduce imidazole 1. After that we introduce benzimidazole 1. And here also we use methyl. After that we use benzyl. So the compounds have been characterized with all the methods, but due to the shortage of space, few are given here. First, let me explain the TG and UV very quickly. As you can see that this is the graph, the weight loss for the compound 1 and 2 is observed at 192 to 193 degrees Celsius, whereas for compound 3 and 4, weight loss is observed at 196 to 200 degrees Celsius. That is the absorption spectra of the compounds. You can see all the peaks are observed from 351 to 407 nanometer, and this is all because of the N to pi star or pi to pi star transition. We have grown beautiful crystal from methanol, and the color of the crystal was yellow. And you can see that the methyl derivative of the imidazole crystallizes as monoclinic with space group P21 by C, whereas the benzimidazole crystallizes as orthorhombic with space group P21-21-21. Theoretical investigation also done in Gaussian software. As you can see that the energy gap for the compound 1 is 4.2 is the highest one, whereas the energy gap for compound 4 is the lowest one, that is the 3.87. It could be explained on the basis of the structure. As you can see that in structure 1 there is less conjugation among these 4, so it shows highest energy gap, but in case of 4 it has the highest conjugation, so it reduces the homolomo energy gap. Now let me explain the biological studies of these compounds. With the little variation of the structure, it shows very different type of biological studies. We have done MTT assay. The psychotoxic activities of the all compounds had been investigated against H520 squamous cell lung carcinoma using MTT assay. The psychotoxic activity was determined according to the dose values of the exposure of the compounds required to reduce survival to 50% compared to the untreated cell. The observed IC50 values for 24 hours reveal that the two compounds exhibit different range of psychotoxicity against H520 cell. The ability of the complex to kill the compounds at 24-hour incubation vary 1, 2, 3 and 4. This is the graph of the compound. The blue one represents the methyl derivative of imidazole, whereas the red one represents the benzyl derivative of benzimidazole. So this is the methyl derivative. In this region it predominates, whereas in this region red one predominates. Now here this is the this is the methyl derivative of benzimidazole, whereas red one is the benzyl derivative of benzimidazole. The methyl derivative predominates everywhere, and you can see this is because of structure. Maybe the benzyl one, that is the benzyl derivative of benzimidazole, it has such a bulky structure, so it could create static hindrance also, and that's why it couldn't be that much effective to kill the cancer cell. The most important characteristic of apoptosis are morphological changes, and it is determined by the acridine orange and ethidium bromide staining. You can see this figure. The cell was treated with this substance 24 hours, underwent both early apoptosis and late apoptosis. The control of viable cell shows this is the green fluorescence and normal cells feature of uniform chromatin with an intact cell membranes, whereas the apoptotic cells showed red nuclei with condensed or fragmented chromatin morphology mostly. The result demonstrated that all substance induce the majority of cell death through apoptosis mode and very fewer in necrosis for 24-hour treatment. To detect the apoptosis at basic level, we adopted phase staining. You can see here, this is the picture of phase staining, which reveal the changes in cytology of the cell with special reference to cytoplasm and nucleus. After treatment with IC50 concentration of the complex for 24 hours, the cells were observed for cytological changes. The observation revealed that the early apoptotic features such as cell shrinkage, chromatin condensation, and fragmentation have seen mostly in treated cell, and small number of necrotic cells were also observed. So it's time to draw the conclusion. In conclusion, we can shortly say that the capability of the compounds to destroy H50 cells at 24 hours is follow this way: 1 2 3 4, and the computational studies also give us the idea about the chemical reactivity also. And you can see this is the reference, and I would like to thank the organizer of NATCOACB 11, and I would also like to thank the Department of Chemistry NEHU for giving me all the facilities. And thank you everyone for your kind attention.